Hello, and welcome to this Nature Research custom webcast titled The Impact of Tissue Processing on Single-Cell Sequencing. My name is Joshan Karpin, and I will be your moderator. Today's webcast is produced by Cytiva. We'll begin the webcast with a presentation from Luciano Martelotto of the University of Melbourne, and then end with a Q&A session. To ask a question, just type it in where it says type your question here, and then press submit at any point during the webcast, and we'll answer them today. And now over to Luciano.
Hello everyone, my name is Luciano Martelotto. I'm the Scientific Director of the Single-Cell Laboratory at Harvard Medical School, and for today I have prepared a discussion on the systematic biases that are introduced during solid tissue processing. The motivation for the topic of this webinar is that the more we learn about solid tissue processing, the more we realize we need to improve it. And what that means is that there is a lot of potential sources of technical and biological variation to consider when planning an experiment for single-cell or single-nucleus RNA-seq. And while some of these sources are obvious to us, even though sometimes we ignore them, the vast majority are unknown. And even if we know them, sometimes we don't understand them. Most of the current single-site technologies attempt to mitigate some of the noise that can accumulate during the multiple runs of PCRs that are usually needed from cDNA amplification to library construction by, for example, incorporating unique molecular identifiers or UMIs during cDNA synthesis. But of course, this doesn't address any changes to the transcriptome that may have occurred and accumulated prior to reverse transcription. As of today, I would argue that pretty much all standard sample preparation methods for solid tissues require some sort of enzymatic and/or mechanical dissociation. And that depending on many factors such as tissue of origin, cellularity, disease state, extracellular matrix contact and composition, fractions of cells of interest, viability of the tissue, preservation of the samples, and others, decisions will have to be made in terms of the type of dissociation, the enzymes to use, the digestion time, and how vigorous the mechanical disruption needs to be at the time of processing. One often forgotten key aspect to consider is the fact that the transcriptional machinery remains active at 37 degrees. That is the typical temperature of collagenase digestion of solid tissues, collagenase being one of the most common enzymes used for tissue dissociation. And as a consequence, the tissue, or more specifically the cells within the tissue, will continue to respond to environmental stress and cues that will in turn introduce gene expression artifacts that have nothing to do with the biology at the time of harvest. Moreover, this tissue digestion is often carried out with suboptimal amounts of nutrients and growth factors, and in the absence of anchorage. Remember, we are generating a cell suspension that is totally artificial. That's not how the cells live in a tissue. And anchorage is a key feature of healthy cells in solid tissue. All of this, together with a harsh dissociation at relatively high temperature, that's 37 degrees, may induce apoptosis or anoikis, and will eventually pollute the viable cell population and consequently generate low-quality cell suspensions. In this webinar, I would like to discuss two of the most important benchmarking studies in terms of sample prep and solid tissue processing. The first paper is from the groups of Saurabh Shah at the Memorial Sloan Kettering Cancer Center in the USA and Samuel Aparicio at the British Columbia Cancer Center in Canada. The other is from the group of Alistair Forrest at the Harry Perkins Institute of Medical Research in Australia. If you haven't done it already, these are must-read papers and I will encourage you to check them out. I would like to highlight that these are not identical papers. They focus on the problem from different angles and consequently they are complementary. I will not go over every single experiment, but I do want to give you an intuition about some of the key findings from both studies that I'm sure will change the way you see solid tissue processing. In this slide, I wanted to show you their experimental designs. As you can appreciate, these are very comprehensive studies with a large collection of extremely well thought out experiments. On the left is the experimental design of the North American teams, involving breast and ovarian cancer tissues from patients and PDX's counterparts, subjected to standard warm collagenase dissociation at 37 degrees and to cold active protease dissociation at 6 degrees. For the latter, they use a serine protease isolated from a Himalayan glacier resident bacterium that had been previously shown to be suitable for dissociation of non-malignant renal tissues at 4 to 6 degrees. Here they also subjected cell lines to treatment with TNF-alpha to induce cell death and to analyze the transcriptome of live, dying, and dead cells. On the right is the experimental design of the Australian team, in which they use mouse kidneys as models to compare and contrast the two tissue dissociation protocols I just mentioned. Those are warm and cold dissociation. Additionally, they also perform comparisons between two single-cell suspension preservation methods. Those are methanol fixation and cryopreservation, and also 3 single nuclear isolation protocols, which I will not discuss here today. On their mission to uncover transcriptional changes in response to the dissociation conditions, the North American team profiled more than 155,000 cells across 48 single-cell experiments, including a range of substrates, cancer types, dissociation temperatures, and tissue states. For all of them, they used the 10x Genomic Chromium V3 platform. The authors started their analysis by examining a set of commonly used quality control metrics across all 48 experiments, including the total number of genes detected, the percentage of transcript mapping to the mitochondrial genome, and the total number of UMI sequence, which are summarized on the figure on the right. Perhaps not unexpectedly, they uncovered significant variability across these metrics. In particular, they observed bi- and trimodal distributions in the percentage of mitochondrial genes across the sample set. It is important to highlight that this variable mitochondrial gene content was also observed in public datasets available from 10x Genomics, where we can see cells with an increase in the percentage of mitochondrial gene content and a lower total number of genes detected. Based on the bi- and trimodal distributions of mitochondrial gene percentages that were observed in the 48 experiments, together with previous assertions about high mitochondrial gene content as being indicative of dead and dying cells, the team sought to determine the contribution of dead and dying cells to the variability observed in the QC metrics. To do that, they treated a non-tumorigenic lymphoblastoma cell line with and without TNF-alpha for 24 hours. The idea behind this was to induce classical cell death pathway before staining the cells with propion iodide and annexin 5 and fact sort the cells into dying, dead, and live populations according to single, double, and negative staining, respectively. Here's the FACS analysis showing the gating strategy. For untreated live cells, PI negative and annexin 5 negative. For TNF-alpha treated dying cells, that is PI positive and negative and annexin 5 positive. And for dead cells, PI positive and annexin 5 negative. Now, the first thing that they noticed was that the cell yield for single-cell RNA-seq data was highly dependent on the cell status, meaning that nearly 8,600 were recovered as live cells based on the staining, but only 1,300 and 900 were recovered as dead and dying cells, respectively, compared to the targeted number, which was around 3,000 cells each. Principal component analysis projections of the 3 cell conditions demonstrated that the cells segregated approximately along the first principal component according to cell status. What is more, the first principal component closely tracked the mitochondrial gene content of the cells, as you can clearly see from the PCI projection colored by the percentage of mitochondrial genes that increases along the PC1. Certainly, the percentage of mitochondrial gene counts was significantly higher in dead cells compared to both dying and live cells. Now, this observation is very important because it justifies the practice of excluding cells with high mitochondrial gene content as being likely dead cells. After observing that the transcriptome of the different cell conditions are not entirely different with each other, they wanted to determine what is the extent of the mixing among transcriptomic states. They also wanted to know whether live cells and dead cells that appear transcriptionally healthy, meaning that they will normally pass QC, are distinguishable. By means of unsupervised clusters of gene expression profiles, the cells grouped into 3 clusters, once again approximately tracking both the first principal component of the data and the percentage of mitochondrial transcriptome. Interestingly, but perhaps not surprisingly, these 3 clusters showed variable composition in terms of cell states, with cluster 1 being comprised mainly of live cells, cluster 2 containing an increased proportion of dying and dead cells, and cluster 3 comprised of mainly dead cells. Furthermore, the percentage of mitochondrial transcriptome was also significantly different between the 3 clusters, with a stepwise increase in proportion moving from cluster 1 to 2 and from 2 to 3. Taken together, these results suggest a model whereby cluster 1 will represent transcriptionally healthy cells. Cluster 2 will represent transcriptionally stressed cells that are likely upregulating stress pathways and have an increased mitochondrial gene content. This can be due to, for example, genome degradation or permeabilization of the cell membrane causing loss of the cytoplasmic messenger RNA, or perhaps an increase in metabolic demand. And cluster 3 represent transcriptionally dead cells, whereby the genome is degraded, most of the RNA has leaked out of the cells, leaving behind mainly mitochondrial transcripts. Next, they wanted to know if within the transcriptionally healthy group, a sorted dead cell can be distinguished from a sorted live cell. So they took cells from the cluster 1, they passed them through a stricter set of QC filter, and then they performed differentially expression analysis between live and dead cells in this group. Of the genes retained for analysis, nearly 10,500 genes, around 20% were found to be differentially expressed. Then, to add a bit of context to this analysis, what they did was to compare this type of intra-cluster variation to the intercluster transcriptomic variation. So they performed a differential expression analysis between cluster 1 and cluster 2, and what they found now was that nearly 81% of the genes were significantly differentially expressed. So taken together, these results show that indeed there are gene expression differences between dead and live cells within the healthy group, that is cluster 1, which supports the idea that the sorted dead cells are somewhat distinguishable from the sorted live cells. However, the magnitude of this variation of expression is relatively small when compared to cells from a truly transcriptionally stressed cluster. Then, they perform a differential expression analysis between 6 and 37 degrees, but this time they combine all the experiments done with the X's and cell lines. In this way, they define a core set of 512 significantly differentially expressed genes, of which 507 were upregulated at 37 degrees and the remaining 5 were downregulated, with an absolute log change of at least 0.5. In the left panel of the figure, you will see the top 40 differentially expressed genes between cells digested at 6 and 37 degrees. This gene set includes multiple canonical stress-related genes such as FOS, FOSP, NR4A1, and heat shock proteins, all of which are known to be induced by collagen digestion at 37 degrees. The panel on the right shows the UMA plots colored by digestion on the top, and then by the normal expression of 3 key stress genes, demonstrating the concordance between temperature and the induction of the stress gene signature. The figure on the right shows a pathway analysis of the different expressed genes, demonstrated that at 37 degrees there is a distinct induction of genes involved in the TNF-alpha signaling via NF-kappa beta, with a remarkable 93 out of the 200 genes annotated for this pathway being found in the 512 core set. Collagen at 37 degrees is usually performed for about 2 hours, whereas only 30 minutes are needed for cold protease. The author wanted to determine whether the gene signature was induced due to the longer incubation time required for complete collagen or due to the enzyme itself. To do that, they conducted a time course experiment in which they incubated breast PDX tissues with collagenase or cold protease for up to 3 hours. The cells were taken at 30 minutes, 1 hour, 2 hours, and 3 hours time points. So what they found was a striking upregulation of the gorgine cell collagenase and cold protease digestion at all incubation times. In their eyes, and also in my eyes, this demonstrates that the choice of dissociation enzyme has a remarkable impact in the cell's transcriptional response, independent of the digestion. However, a subset of the core gene set was further upregulated with increasing digestion times under collagenase digestion. To quantify this, they performed several transcriptome-wide pairwise differential expression analyses that I will briefly explain. Here you have a comparison between the log-fold changes at 30 minutes and 2 hours digestion when only using collagenase, where 420 of the 512 stress genes were induced, as compared to 184 of these genes being altered when using only cold protease. These two panels show a comparison between collagenase and cold protease digestion at 30 minutes on the left and 2 hours on the right, highlighting a time-dependent increase of the gene express signature induction. These results very clearly demonstrate that both digestion time and digestion method contribute to the transcriptomic stress response. Finally, this panel shows that a highly similar set of genes are affected by both digestion time and digestion method, with a large correlation between the log-fold changes of contrasting 2 hours to 30 minutes digestion using collagenase only, as compared to a collagenase versus cold protease digestion at 30 minutes. These results suggest that the cellular response to the stress generated due to the different factors involved during the tissue processing, such as temperature and time, converge to a common set of pathways. Just briefly here, and to show you some nice human data, the panel on the left shows the top 40 differentially expressed genes between collagenase and co-protease protein from previous analysis in PDXs across each cell type within breast and cancer patient samples. What they found, and what we can see from the figure on the left, is that there is a consistent upregulation of stress genes, with up to 78% of the 512 stress genes being upregulated across all cell types, and with 9 to 55 of them found significantly upregulated depending on the cell type. The panel on the right is a global pathway analysis of the differentially expressed genes for each cell type, showing a conserved upregulation of NF-kappa beta signaling, apoptosis, and inflammatory pathway as the most upregulated in all cell types. Now it's the turn of the estrogen, whose work, as I said before, covers not only sample dissociation but also storage methods and comparison between single-nuclear RNA-seq and single-cell RNA-seq using the 10x genomics platform. Here, we will only discuss the experiments related to tissue dissociation, but I will strongly encourage you to pull paper out because it's pretty amazing work. So the first thing that they did was to perform standard bulk RNA-seq profiling of single-cell suspensions to find the stress response genes in warm-dissociated samples. Differential expression analysis of these identified 71 genes with higher expressions in warm-dissociated kidneys and 5 genes with higher expressions in cold-dissociated kidneys. When looking at the gene ontology, the top significantly enriched biological process in warm-dissociated kidneys was regulation of cell death. This included immediate early genes such as FOS, FOSB, JUN, and JUNB, and the heat shock proteins, which is in line with the previous study we just discussed, and also confirmed that the warm dissociation of tissues induces substantial stress-related transcriptional changes. What they did next was to characterize the differences between the two tissue dissociation protocols by single-cell RNA-seq profiling using fresh cell suspensions. The panel on the left shows the number of differentially expressed genes between cold and warm dissociation for each one of the cell types identified in kidney samples. The differential expression analysis found 64 genes as overexpressed in warm-dissociated tissue in at least one cell type. And once again, the gene ontology analysis reported regulation of cell death as one of the top significantly enriched terms. The panel on the right shows the stress score for each cell type. And here the stress score is based on the expression of a set of 17 stress response-related genes that were found in the bulk analysis. As you can see from this absolutely striking figure, the genes most commonly overexpressed in warm-dissociated samples across cell populations include immediate early response genes, such as JUNB and JUND, which were differentially expressed in 7 out of the 14 cell types identified in kidney, and JUN and FOS differentially expressed in 5 out of the 14 cell types. And finally, something remarkable that came up from their analysis was that several cell populations were less abundant in warm-dissociated kidneys in comparison to cold-dissociated ones, including, for example, podocytes, mesangial cells, and endothelial cells. Not surprisingly, these depleted cell populations also show significantly higher expression of the stress response-related genes, which would suggest that these populations are sensitive to warm dissociation and consequently underrepresented. So to conclude, few take-home messages that will be great for you to remember next time that you plan your single-cell RNA-seq experiments using solid tissues. So the first one, of course, is that the choice of methods and conditions for tissue dissociations are very important. And this is because they are highly likely to influence the cell yield, viability, and transcriptome state of your sample. Now, the magnitude of that influence, of that impact on the transcriptome is both tissue and cell type dependent. What was clear from these two papers is that there is a large variability in the QC metrics and that there is no key metric that is good on its own. In an ideal scenario, each tissue sample and dissociation method should be assessed for dissociation-induced signatures before undertaking any large-scale single-cell RNA-seq experiments. For any given tissue, all cells exhibit some stress in response to dissociation that is both temperature and time dependent, with some cells even exhibiting cell type specific responses. And finally, and the highlight of these two spectacular studies is that the stress responses, which may significantly influence the interpretation of your single-cell RNA-seq data, are minimized by dissociation at cold temperatures. I hope that with these two amazing benchmarking studies, I managed to convince you about the importance of good solid tissue dissociation techniques to the overall process. And this is not just only for single-cell or single-nuclear RNA, but also for any of the other single-cell genomic subjects. I know that it's quite challenging sometimes to get the right conditions in terms of sample prep that is suitable for every single-cell application. So new technologies can make sample processing gentler, faster, standardized, and perhaps more generalizable. On that final note, I want to thank Sidiva for the invitation and also the NatureComms team for helping us set up the webinar. And of course, a very special thanks to all the attendees for listening. Please feel free to reach out directly via or to my email address that you can find on the screen. Thank you.
Thank you for your presentation, Luciano. It is now time for the Q&A. Ask Luciano a question, just type it in where it says type your question here and then press submit. So Luciano, our first question, and this one asks, will automated tissue disaggregation really help address the high throughput demands being placed on single cell sequencing?
Oh, yeah, absolutely. Look, I think it's the aggregation platform for either fresh or frozen type of samples. New opportunities for any project that is in large scale. Projects like the Human Cell Atlas and not even that of that scale, even smaller than that. I think a dissociation procedure that involves a dissociator, it can, might well benefit from that sort of standardization in the processing.
Excellent. Thanks, Luciano. Our next question asks, what are the possible reasons for genes overexpressed in the cold protocol? I think they mentioned that, as the authors of the paper stated, 6 degrees is usually the temperature that shuts down all transcriptional and translational machinery. Is that something you can sort of help answer?
So that's a very good question. They don't really explain exactly what the reason of the induction of those particular genes. I think what we need to consider, this is just a differential expression, and we need to consider that probably those genes are expressed as or at least differentially expressed, because while you're putting the samples on in cold dissociation, the machinery is still active. So cold also induces stresses. There are evidence in the literature that cold actually can induce stress genes because it's not the normal status of the cell. So probably that it's happening now. 4 degrees, the transcription machinery, the activity is lower, but it's not completely obliterated, right? So, and I think that expression of those genes may have to do with some sort of vestigial activity, or it can be that those genes are activated while the temperature is going down as well.
Excellent. Thanks, Luciano. Our next question asks, do you encounter challenges with stability as a result of your current tissue disaggregation approach, or the approach used in your sort of research department?
Yeah, I mean, we in previous work we always have with stability, but it's it had to do a lot with how this sample was treated. In the sense, I always ask people to treat the samples nicely, in the sense that they need to to gather as much information as they can from those samples. So by the time they bring it to me, I know exactly what to expect from those samples. So some tissues are more sensitive than others to temperature changes. Some others are more resilient. I think at the moment, the common practice is to put the cells in a media and condition in a media that the cells are happy. It could be a growth media. If you know about that, it be, it could be any FBS. The cells in a condition that resembles where they're coming with a lot of nutrients, but also we have to think about that is sort of an artificial state, and that is not going to last for too long. By the time we start dissociating, some of the enzyme will not be compatible with some of the components of the FBS of the serum, so they will not be active enough. So what you need to do in those cases is to remove the serum and treat the samples in a medium with low serum. So, and that is what I was mentioning before during the presentation, that the amount of nutrients is very low, and you are doing manual mechanical dissociation, and on top of that, you are treating aggregating the extracellular matrix, so the cell lose that ability to interact with other cells. All of that contribute to cell death. So there is very little that we can do in that sense, because every tissue is different. So that it's so my advice to every person that is coming with a new sample is try to know as much as you can of those samples, especially in the way, try to preserve the way, if you're going to transport those samples, try to preserve those samples in the way that they feel most happier. But sometimes it's really difficult to find those conditions. So it's always experience and trial and will actually give you the best conditions. There are plenty of protocols available in Protocol CEO and in the Human Cell Atlas that you can start with. Don't make the first dissociation attempt your first single cell experiments, because it's probably doomed to failure. So practice, make a pilot of your sample dissociation, and then once you get that 100% nailed down, then you can move to the next stage. And again, not all dissociation methods work for everything. Protease has worked well for these tissues that I just mentioned, and other tissues that are not necessarily published, but some other tissues are more resilient than to cold protease. So, and that is probably where standardization conditions for dissociation and where dissociators can actually be quite beneficial.
Excellent. Thanks, Luciano. Our next question, do you have any insights into using extended cold protease digestion? For example, overnight digestion for tough tissue samples?
Yeah, I've seen people doing that. I don't have particular insight myself, because I've not done it. But I think it's one of these things that you have to try to know whether that works for your sample type. I think it's an extremely important piece of information. So if someone actually does, it would be good to put it out there, because it's a very useful experimental condition to try out.
Excellent. Thanks, Luciano. Our next question. Does the stress genes influence the overall cell states? And is it possible to perform regression to remove the effects of cell stress during normalization or DE testing?
Yeah, that's a very good question. So essentially what you're going to have is you're going to have a set of genes that have been induced during this processing time that may not have anything to do with the biology that you were actually studying when you collected that tissue. Okay, so it may actually be easy to recognize, and you will be able to remove them, okay, by regression analysis. That's what people mostly do at the time. But in other situations, those genes that are induced are more tricky to remove, because probably you're depend on the question, right? So if your question is about, okay, I'm studying cells under treatment, and you know that treatment is going to induce some stress. So if you also have stress induced by collagenase, what is the signal and what is the noise? Okay, so that's a confounding effect. So you probably should do a pilot study, and I always insist on pilot studies, because it gives you a lot of information. And for you to do a more educated approach for the next step. So if you do a pilot and you see that there are some genes that are not supposed to be there, like genes that, for example, this group of genes that we discussed today, and they're usually the same type of genes. And if you see them, just make sure that you know that they exist, that you know that your method is actually inducing them, so you're able to regress them out properly. Because otherwise, you may be regressing something that is part of your biology.
Excellent. Thanks, Luciano. And just a quick reminder to our live audience, you still have time to ask a question. All you need to do is just type it in where it says type your question here and then press submit. So our next question, Luciano, and this one asks, is there any data on stress signatures in protocols using mechanical and enzymatic disassociation for shorter times? And are there differences between warm and cold conditions? Is that something you can help answer?
Yeah, I haven't seen anything for shorter time than 30 minutes, but maybe this, and at least not published, that I have seen. I think it's probably the shortest time that I've seen is in this case that it was just 30 minutes, which I think is pretty short for everybody that is used to work with tissue processing or dissociation with the standard enzyme. 30 minutes is quite a short period. I haven't seen anything lower than that.
Excellent. Thanks, Luciano. And next question, I think, picks up on sort of one of the answers that you gave previously. And this one asks, would keeping the cells in a happier medium while running through the cell platform improve data quality?
Look, yes, we've seen situations. I'll give you an example. So we had, but again, this is always depends on the question. So you need that the question actually makes sense for what you're trying to do, and also whether this happy, media is actually compatible with the platform. So there were a few cases in which we were working with a cell type that came from an organism that lives in very high salt water. Okay. So the moment that you put the cells in PBS and BSA, the cells would die. And we can see that clearly in the data. So the cells would die. So what we had to do is just to sort of modify our protocol to keep the cells in this high salt containing media right until the end in the platform. So that's not something that can be done with any platform. It can be done with platforms in which you can actually modify the microfluidics to be able to dilute the contents of salts in the drop, or it could be done also in plates. But it's in this case, if we didn't keep the cells in a happy state, our data was completely rubbish, to be honest. So that it was a typical example in which knowing the conditions in which your cells are happier, and knowing that those conditions in which your cells are happy were incompatible with the platform that we were using, in this case was a droplet 3D platform. So we need to help us to sort of engineer and design to make the single cell approach possible for this. All right. So, and I think the similar idea can be applied for any cell type. As long as you know and you have some sort of background about the composition of the media, whether that is compatible with the platform that you're using for single cell, and whether the cells can be kept happier in another media. If in the case that the media is incompatible, what is the other alternative? I think if we think hard enough, we can find the conditions to make the experiment.
Excellent. Thanks, Luciano. Our next question asks, can we delay the degradation of the transcriptome by reducing the processing time?
Yes. Well, ideally, most of these things things that we're discussing at the moment, we are not adding this to RNA degradation. Okay, it could be that is happening. So usually you see as a signal, it could be RNA degradation plus the activation of new genes, right? So or speed up turnover of the messenger RNAs, and that's what you start seeing those differences. So the differences in your transcriptome can be due to new de novo activation of genes, can be to an enhanced turnover or reduced turnover of the messenger RNA for a particular species of or it could be actually degradation per se. Those experiments, you include quite powerful RNase inhibitors, which are good, but not, they are not 100% bulletproof. So to answer your question is yes, if you delay or you make your processing faster, it's always better than slower. So 30 minutes is better than 2. But I think the key here is not just only that, it's also the way that you handle the tissue prior to the processing, the way that you cryopreserve, the way that you dissociate the tissue, your mechanical forces, the type of enzyme, the temperature, okay, and that what is the media that the cells will suffer least while you're doing all this, mistreatment.
Excellent. Thanks, Luciano. And sort of following on from that, next question asks, what stage does the degradation of mRNA start? And would you say that the more time taken to digest the tissue, the more the chance of actually sequencing artifacts?
Look, by default, and this is something that I like to think for myself, it works for me, but I think I always consider that the RNA starts degrading from the moment that the tissue is out of the body. Or if it is a cell culture, the moment that you take this out of the normal condition. So, and this is, again, degradation happens there. Whether that degradation is actually significant. It may be significant for some cell types. If you have a tissue that is very heterogeneous, it may be more sensitive, or maybe more the effect of that degradation can happen in different cells of that tissue to a different rate, and that is because the cytosol content is different, right? So from cell type to cell type. So all of those things is a lot of variables, and I think all of them contribute to that differences. As you saw in the experiment from Denisenko, they see differences in the enumeration of some of those cells, and that thing that had to do because those cells it's not that they cannot detect those cells because the cells were not there. It's just because they did not pass the QC, and then you're just removing them. So why is that? That can be because the cells started dying, the plasma membrane became more leaky, and then RNA came out. Then you have a high material content, and then you just remove that in your default pipeline. But it also could be that the RNA was more degraded in those cells for a particular reason. I can't enumerate all the reasons that are possible, but my suggestion is to think that the RNA starts degrading from the moment that you start working on the sample. So that's why the faster, the less number of steps you do, and the more gentle you are with the sample, the better it is.
Excellent. Thanks, Luciano. Our next question asks, would a single nuclei approach deviate a little of the cell death issue? How would it compare to the single cell approach in general? Is that something you can help answer?
Yeah. Look, I think probably 90% of all the single cell genomics that I've done is single nuclear RNA-seq. And the reason why we've done that is because single nuclear RNA-seq, it's not necessarily the topic of this. It would have been great for me to include that, but I think a single nuclear RNA-seq allows you to expand this ability to do single cell genomics to samples sometimes are more difficult to process. For example, if you have any, for samples that are snap frozen or flash frozen, it's very unlikely that you're going to have large populations of high viability, which is essentially is one of the essential parts of single cell genomics, especially for those that we are, because especially for droplet microfluidics. It's also very important for plate-based, but for droplet microfluidics that is important, because in those situations, a cell that is damaged or that is dead, the plasma membrane becomes very leaky, the RNA comes out, and then it gets, it becomes part of your reverse transcription buffer where the cells are going to be, dissolved in, and then when you capture that in a droplet, what you're going to see is a lot of contaminating RNA, what is called as ambient RNA, and that's a bad thing to have. So in those situations, single nuclear RNA-seq is the way to go. Is the way that I will always suggest to go. In some situations, for example, when you have a post-mortem material, I had situations in which I have received some samples. I say, okay, this is a post-mortem material and it's been cryopreserved. Okay. Now, when someone tells you cryopreserved, the first thing that you think is, okay, this has been put in a media that is a cryoprotectant and it's been slowly frozen and viability preserved. Now, what you need to think is that there is a time between death and biopsy. So in some of these situations in which I received those cryopreserved samples between, quotation marks, the RNA was really, the cDNA showed me that the RNA was really bad quality, very, very degraded. So, but they would have sworn to me that was cryopreserved. But then if you start asking a little bit of question, that comes from a post-mortem tissue that comes from a patient that died the day before. Okay, so the sample had been in at least a room, not room temperature, but say 12 degrees for nearly 10 hours. So that it's something that it's problematic. So post-mortem is a type of tissue that is very tricky if you do not have background information. Also, as soon as the body dies, or either from an animal model or from a patient, there is a lot of decay that happens very quickly. So in those sometimes can help. Okay, that doesn't mean that you cannot do a single cell RNA-seq if you have a lot of, if all the biopsies were taken very quickly after. Okay, and in situations in which you suspect any type of viability issue, single nuclear RNA-seq will allow you to do that. If you see that when you do some enzymatic dissociation of, even from fresh tissue, you will see that some of, after with experience, you will see that some tissues dissociate better than others. And within a tissue, some regions of the tissue may dissociate better than others. Because at the end of the day, what we're trying to do is try to break the extracellular matrix. And the matrix can be different in different parts of the tissue. Okay? So what you are doing is, in cases, is you're treating the tissue as all of the cells in the tissue are surrounded by the same type of extracellular matrix, and they all need the same time of dissociation, even if you do some mechanical. So you end up some populations within that tissue dissociating poorly, and then when you take the single cell suspension, you are biasing your cell population because the cell type that didn't dissociate very well, you get lost you lost that as a debris. Okay. In those situations, if you think that you have very unbalanced dissociation efficiency within your tissue, single nuclei can also help. And so on. So there are a lot of things in which nuclei can be very beneficial. And there is also, of course, that you don't have to worry about any enzymatic control here. Nothing. This is always done, at cold temperature. There is no activation of the transcription machinery. Is all done very, very quickly, and it's a very short processing. Now, what's the other side of the coin is that it's less than 1% of the total RNA is within the nuclei. So in some situations, There are some approaches that are really hard to do. If you do single nuclear RNA-seq just for gene expression analysis, it's very good. It works very well. There are plenty of protocols to actually get it to work very nicely. Some involve FACS, some other involve other methods to remove some of the debris. But if you got to do something like TCR sequencing, for example, in my hands, and after I try a million things, it does not work. And that is because the TCR or PCR transcripts are very lowly expressed, and whatever you find there is just a lot of background. So again, it works for many things, but also has some limitations and some challenges. But I would say that if you have any issues with viability, nuclear is the next step, and it could be a savior of your project.
Excellent. Thanks, Luciano. Next question, I think, is something that you touched upon in your answer just now, but it asks, how important is it to use the fresh tissue for single-cell sequencing compared to fresh frozen tissue?
I think a fresh tissue is the ground truth. Okay, that's the. But again, we are assuming that here that the fresh tissue has not been stressed. Okay, again, the moment that you take the tissue out of the organism, you assume that the machinery is still working, even if you put it the transcription machinery is still working, even if you put it at 4 degrees. So, but let's assume the situation, and that you can find the right cocktail of enzymes to dissociate the entire tissue into single cell. I think that's the best. That's the ground truth. That's what you want to know. Unfortunately, we don't have the way to do single cell transcriptomic in situ. I'm hoping that some of these ginormous companies can do that for us in an easy way. But at the moment, we are depending on taking the sample out of the organism and dissociate that. All right. So I think that's a good way. If I had to choose how I would do it, I would do fresh, and then I would do cryopreserved sample in the proper way, with a cryoprotectant, and then slow freezing, and then you just long-term lower temperature. And then if nothing of that is possible, I will probably do nuclear. Some people will say, okay, what do you think about methanol? If you think about that question and say, okay, for me, methanol it's an option, but I have not had much of a great success. I know that it works for some cell types, but we ended up getting a lot of the cells that get very sticky, and it's just really hard. I don't think it's standardized enough.
Excellent. Thanks, Luciano. Well, I'm afraid that is all we have time for today. Before you go, we would really appreciate if you could fill in a very quick survey about the webcast. I would now like to thank the speaker, Luciano Martelotto, for today's presentation and for answering your questions. I would also like to thank the webcast sponsors, Cytiva, and of course you, the audience, for taking time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com webcasts. Thanks for watching, and I hope you'll join us again soon.